Electrophoresis is a method of separation and analysis of macromolecules, DNA, RNA, and proteins. For this practice, the molecules we are going to analyze are DNA from cedron tissue and human blood. Electrophoresis is used to estimate the size of the DNA fragments, also to identify, quantify, and purify them. This happens by applying an electric field to move the negatively charged molecules through a matrix of agarose, acrylamide gel, or other substances. The shorter molecules move faster and migrate farther than longer ones, because short molecules migrate more easily through the pores of the gel. This resulting in separation based on size. In genetic engineering is a basic procedure because of its importance in DNA analysis.
As mentioned before, in this practice, we're going to follow the laboratory handout to prepare an agarose matrix at 1% to run the electrophoresis of the DNA samples from cedron tissue and human blood previously extracted.
The materials used in this practice are agarose, DNA samples, DNA size standard, electrophoresis buffer TAE, gel Louding buffer, bromophenol blue, and silent cyanol. Electrophoresis chamber, gel sealing tape, Erlenmeyer flask, transient illuminator, UV lights, paper towels, microwaves, and a power supply. The agarose gel serves as a matrix of the phosphate molecules that make up the backbone of the DNA molecules that have a high negative charge migrate toward the positive end of the field. The DNA samples are from cedron tissue, which is a plant, and human blood. Also, a letter of DNA size standard is needed for having a guidance for knowing the size. The electrophoresis buffer, TAE, which is composed of tris, which is a strong base, acetate, that is a strong acid, and EDTA. This buffer creates a slightly alkaline condition where DNA is protected against hydrolysis, and it's also used to maintain an ideal pH. The gel Louding buffer composed of bromophenol blue, which colors the sample bright blue, so that it's easy to track the process through the gel, and silent cyanol, which is also used as a color marker. The UV transilluminator is used to view the migration in the electrophoresis gel, and the power supply is for the purpose of the migration.
For the preparations of the electrophoresis buffer, 0.4 grams of agarose were weighed in order to prepare solution of 0.8% agarose concentration. The electrophoresis plate was prepared. First of all, the cassette was put on a horizontal place. In order to place the acrylic plate into the cassette, the movable wall was set into the appropriate size for the acrylic plate. After that, with the help of the leveling bubble, the cassette was leveled, making sure that the bubble was in the middle of the leveling bubble and moving it with the help of the leveling feet that are in the cassette. The cone was placed in the level cassette inside the acrylic plate. For the preparation of the agarose, for the electrophoresis, the agarose powder was emptied into an Erlenmeyer flask.
Then, with the help of the cylinder, 50 milliliters of TAE 1 times buffer was measured. Next, the buffer was poured off from the cylinder into the Erlenmeyer flask where the agarose was already in. Then, the flask was submitted to heat inside the microwave oven for 30 seconds, taking care of not letting the buffer get into its boiling point. After this, the flask was taken out in order to mix the solution by swirling. These last two steps were repeated until the agarose was completely dissolved. The next step was to cool down the agarose and, before it solidified, 5 microliters of ethidium bromide were added with the micropipette. Then, the flask was submitted to circular movements in order to mix the prepared solution. After this, the prepared solution was poured into the acrylic cassette. It was done by the bottom corner in order to have a uniform gel.
The agarose solution was left to get cooled down and solidify. Then, the electrophoresis tank were put horizontally on the table with open lids and the cables were connected into the power source. After that, it was filled with TAE buffer until it reached the cisterns. Once the gel was ready, the comb was pulled out, leaving the small slots. Then, the acrylic plate was taken up from the cassette and it was placed inside the electrophoresis tank. Once the agarose gel was inside the electrophoresis tank, it was filled up to its maximum volume. 10 microliters of the DNA sample from plants were mixed with 3 microliters of the loading buffer, and 10 microliters of DNA sample from blood were mixed with 2 microliters of loading buffer too. Then the size standards were loaded into the slot from both sides of the gel. In order to put the samples into the slots, the micropipette was loaded with DNA sample and it was carefully dropped into the submerged gel wells. The lid was closed, the power source turned on, and the voltage of 100 volts was set. After 45 minutes, the electric current was turned off, the lid opened, and the gel taken out. The gel was submitted into the UV light in the transilluminator in order to see the band patterns. Finally, a photograph was taken with the use of a computer connected into a camera inside the transilluminator.
According to Green and Sandbrook, agarose is a linear polymer composed of alternating residues of D-galactose and L-galactose joined by an alpha and beta glycosidic linkages. The main use of this polysaccharide in any molecular biology laboratory is for agarose gel electrophoresis. As Smithis and collaborators state, electrophoresis is a separation method frequently used to analyze DNA fragments by absence, presence, and its different sizes. Because there are many phosphates in the DNA structure, it is usually negative charge. With these characteristics, the DNA samples are put in agarose and an electrical field is applied to separate the different sizes of DNAs. DNA fragments with smaller sizes can run faster in the gel, while DNA fragments with larger sizes only run a short distance. During this practice, the protocol was extracted from Green and Sandbrook, with minimal variations according to the materials and equipment available in the laboratory. Because this was our first time doing the electrophoresis protocol, some mistakes were observed. As Curran and Schofield state, there are several common mistakes during the first gel electrophoresis. For example, the voltage and time set for the technique, the amount and how the samples are loaded into the gel. None of these errors were present except for the loading of our samples into the gel. Samples 1 pm and 1 vm skip one slot between them. After the stage procedure, a transilluminator was used to reveal the bands. On the 1% agarose gel, there were no DNA bands, and on the 0.8% agarose gel, 5 bands were noticeable. Our team's band did not appear on the gel, probably because of an error during both the plant and mammalian DNA extractions. This could be due to the incorrect removal of the supernatant, carrying with it most of the DNA.
Another reason that could affect our electrophoresis result was that in the case of DNA extracted from citron, more TE buffer was added because of an incorrect usage of the micropipette. This led to an important dilution of our samples. Electrophoresis is one of the most important techniques used by molecular biologists. To name only a few applications, DNA electrophoresis is used to map the order of restriction fragments within chromosomes, to analyze DNA variation within a population by restriction fragment length polymorphism, and to determine the nucleotide sequence of a piece of DNA. Because of that, nowadays it's difficult to imagine a genetic engineering protocol without the usage of this method.